All right, we're doing Unit 2, Lecture 2 with environmental adaptations. So continuing on with Chapter 9, we're going to start off with a concept based around E. coli. And with E. coli, what we're going to discuss is the fact that as a living organism, E. coli can essentially take in raw ingredients and reshape, break down, and reform those ingredients to essentially produce all the compounds inside of itself. This happens to be a well-studied organism. All organisms do this to a certain extent. E. coli happens to be very good at it. But what this is implying is that E. coli can make around 5,000 different compounds. Most of these are things like proteins. You have a variety of nucleic acids, carbohydrates, lipids. However, to make all those 5,000 different compounds, E. coli only needs about 10 different types of nutrients. So that means with 10 raw ingredients, you can essentially come up with roughly 5,000 different compounds. It would be similar to having something like flour, eggs, milk, baking soda, etc. in your kitchen, and from roughly 10 ingredients, you could make many, many different meals or many different recipes. So the idea then is that these ingredients have to be acquired and or synthesized in some way. So a growth factor is something that a organism requires but is unable to be synthesized. So looking back at that previous slide there, those 10 compounds that E. coli needs, those would be considered growth factors. What they are is something up for more discussion, but you need roughly the basic building blocks, in this case growth factors, things that you have to get from your environment. Once you have those, you can then synthesize what you don't have and fill in all the missing gaps. And so E. coli will do this with both anabolic and catabolic enzymes. These are enzymes that can build things up and or break things down. And growth factors are those requirements in order to fill all those needs. Usually these have to be obtained from the environment in some sort of complete molecular form. Examples of these include, but are not limited to, things like amino acids, vitamins, and even nucleic acids. In humans, for example, you have essential and non-essential amino acids, as an example. And in humans, roughly half of the amino acids out of the 20 you can naturally synthesize yourself, and then the other half, roughly, you have to acquire from your diet. So essential amino acids in the human diet are those amino acids that you have to acquire through your diet because you can't make them. The other half are considered non-essential, and you can build those with any kind of general nutrients that you acquire. Now, when you start looking at what organisms do with those nutrients, you can describe those characteristics using different terms. For example, heterotroph, autotroph, photoautotroph, chemoheterotroph. Here we're going to be discussing the chemoheterotrophs. In this situation, what organisms do is they'll acquire these growth factors and these compounds from other types of living organisms. Okay, and so they have to basically go out and find organic compounds that are already existing. And from there, they'll use those for building blocks and also as ways to make energy, which is what is called ATP. Once the organisms acquire all the nutrients that they need, they'll start to use some of those to build, and then they'll start to break down some of those for energy. Those that can do that with oxygen are known to exhibit aerobic respiration, or respiration that is aerobic. Organisms can also do this in the absence of oxygen, and this is often referred to as fermentation. And so depending on the organism, with or without oxygen, organic compounds will be broken down to make ATP and used as building blocks. Now, when you start looking at how organisms play a role in the larger context, they're getting their nutrients, they're acquiring them, what is happening sort of downstream from that? There's one group of organisms that comes up that are really important, and these are referred to as saprobes, sometimes called saprophytes or saprobic microorganisms, as the title here shows. And what they will do is occupy a niche as decomposers. So they're considered chemoheterotrophs. They acquire their carbon from organic compounds in the environment. But here, these types of organisms will literally get those carbon compounds from decomposing organic matter. And what they do is they release enzymes, as we'll discuss here in a little bit what that looks like, into their environment to break decaying matter down. And that is how they acquire their carbon and ultimately produce their energy. These largely are considered, the most common organisms that do this are fungi and bacteria. And these are important when I say downstream, what happens from the metabolic processes. Saprobes are important because they ultimately kind of recycle nutrients in their environment. And we'll come back to that here shortly. Now, there are obligate saprobes. They only exist on dead decaying matter. And then there are facultative parasites, which are essentially organisms that can live in the environment or become a parasite and cause infection in a living organism. Okay? These are sometimes referred to as opportunistic parasites. As we'll see here in a minute, then we can also see what are called obligate parasites. So basically, when you start looking at what happens downstream from how you acquire your energy, you can do it strictly by feeding off dead organic matter. And in that regard, you're considered a saprobe, but in this case, where you're strictly doing it, you're considered an obligate saprobe. And the effect of that is usually just that you play a role in the environment in sort of recycling and breaking down rotting material, which is generally considered a good thing. Facultative parasites are most likely to be found in a similar kind of role as obligate saprobes. The difference is they can also jump into a living organism and become a parasite. So facultative kind of means either or, and that's where the term opportunistic parasite comes up. Now this is a broad spectrum of different examples here, but some examples in humans would include things like Candida albicans, which is a common cause of yeast infection, Staphylococcus aureus, which is a skin bacteria that can ultimately cause infections. In fact, MRSA, which is a methicillin-resistant Staphylococcus aureus, is commonly associated with this microbe. Pseudomonas aeruginosa is another pretty common opportunistic infection pathogen, one of the more common ones that affects people with weakened immune systems. And Streptococcus pneumoniae is similar. One of the most common reasons that you get secondary bacterial pneumonia during a viral infection. Viruses come into the lungs, weaken and damage the lung tissue, and make you susceptible to a secondary infection, Streptococcus pneumoniae, which is more likely to cause infection given the opportunity that a viral infection imposes. So those are some examples there of facultative parasites. Now, an obligate parasite then is one that can only live in a host, and so we start to call these strict pathogens, would be another way to describe it. Obligate parasite is the term used here. And then you have an even more specific category called obligate intracellular parasites. And so these are organisms that simply not only live inside, or not only are required to be a parasite, but are actually parasitize the inside of their host cells. So viruses are a good example of this, bacteria, and even protista. So for example, there would be an example of a protista would be those that cause malaria, the malaria-causing protista. And then you also have, like with bacteria, certain tick-borne diseases fall into this category. So these are often rickettsia, tick diseases, things like Rocky Mountain spotted fever is an example. Ehrlichia is another example. And so these live inside of your cells. Okay, now when you start looking, as I mentioned, kind of the downstream effect of what's happening, you're acquiring energy from carbon as a heterotroph or a chemoheterotroph, okay? You can do that from dead rotting material, you can do that from living things, and you can kind of do it one way or the other strictly, or kind of go back and forth. One way or the other, when you start looking at the organisms that are usually doing this, they're typically bacteria and fungi. And one of the things about these organisms is as it says here, they have rigid cell walls, and those cell walls are really unable to absorb large structures. And so as a result of this, they end up being unable to absorb anything relatively large into their cells. And so as a result, they actually release digestive enzymes, and those digestive enzymes are going to break things down on the outside of the cells. And as a result of that, these macromolecules break down into smaller and smaller pieces called monomers. And monomers then can be transported into the cell. And from there, then the organism can break it down and use it further for energy or building blocks. Okay, but the reality is the process looks something like this. The organism could be a bacteria or a fungi here. This looks more like a bacteria. You have relatively large organic nutrients. They can't be absorbed across the cell due to the rigidity. And as a result, enzymes are released, the enzymes break down the matter outside the cell. And as they break it down, it's then slowly absorbed back into the cell in tiny little amounts. Okay, and so these steps here go through that. Substrate available, enzymes are transported outside, enzymes break it down, smaller, smaller, and smaller. Tiny little pieces now are available and can be brought back into the cell. Now, when you start looking at the downstream effect from the way nutrients are acquired, this can actually sort of help you understand how this plays, how a saprobe and or a parasite ends up causing either something positive or something negative. So for saprobes, these extracellular digestive enzymes, they're breaking down decaying matter, allowing the organism to get energy. And in the general sense of things, this decaying matter is really just dead, rotting vegetation in most cases. Could be dead animals, dead carcasses, could be fecal waste and things like that. It's generally things that are considered non-usable and, if nothing else, an obstacle. And so by breaking it down, generally speaking, these organisms are doing something good for the environment, okay? And in part of that, they're actually releasing an excess amount of these molecules that they can't fully absorb back into the environment. And so while they're trying to feed, they're actually releasing nutrients all around them at the same time. So I kind of think of this as a messy eater and someone who's eating too quickly and can't get all the food in their face or something like that. And as a result, there's just food going all over the floor. Okay, and that food now can be eaten by some kind of animal that comes along. And so imagine you're eating outside and food's going all over the ground, and now you're sort of feeding whatever animals come along to eat it. That's kind of how I look at this from a metaphorical standpoint. These organisms are eating and making a mess, and that mess ends up being beneficial because the other organisms around them can feed off of the scraps, so to speak, that these organisms throw off. And so this ultimately ends up benefiting. It's generally considered a good thing in most cases. And as a result, these are referred to as mutualistic symbiotic relationships, which we'll define those at the very end in a little more detail, or at least reference to where to look at those in the book. But that's a type of relationship in which one organism kind of benefits another. So the saprobes tend to benefit other organisms around them. And although there's many, many specific types of relationships here, these tend to benefit things like plants and algae overall. So here you can see a figure of a fungus. This would be like a part of a fungal cell right here. Here you can see some of the organelles, Golgi apparatus. Here you can see the cell membrane. And here what's happening is the cell is releasing digestive enzymes. And you can pause this here and read all of these to help you better understand this figure. But the intracellular structures are being released outside. In this case, we're looking at digestive enzymes, extracellular enzymes, okay? And then those enzymes break down the larger particles, as I showed in the previous figure. Now the small organic molecules that are able to be absorbed are brought into the cell and can actually be utilized in the cell for nutrients and building blocks. And that process repeats. And then from a broader standpoint, in the soil, these microorganisms essentially do this in proximity to things like trees. And this would play a role in algae as well. I don't have that in the figure here. But in the soil, where all these organisms are releasing some of these nutrients, those nutrients are being picked up by things like tree roots. That is a type of energy and set of growth factors for various forms of trees, plants, grasses, things like that. Those grow, and then they can give off beneficial byproducts to other creatures. I mean, for example, they can feed a rabbit, and then a rabbit can feed a fox, and so on. So ultimately, in the soil, very often, these decomposition processes are really benefiting everything in the environment. Although it's not exactly obvious on the surface, once scientists start investigating this, they realize all the way down into the soil, you have this community of organisms that are benefiting the entire ecosystem around them. So saprobes, in the way that they end up decomposing, are actually beneficial. Now, parasites essentially break down organic matter in the same way, but in the context that they do it, these end up forming parasitic symbiotic relationships, which is pretty much the opposite of a mutualistic one. And what happens here is that the parasite functions in a similar way to the saprobe, but the parasite is the only one that benefits. And that's why it's considered parasitic. Okay? We often refer to these as pathogens. The difference often depends on context. So when you start looking at, okay, well, what is the difference then? The parasite is going to release enzymes that actually cause tissue damage in living organisms. And so they actually have specific types of enzymes that are capable of reacting in body tissues and actually using living organisms as a source of nutrients. And so you can actually see that one of the characteristic features of all parasites, all pathogens to some extent, is that they have to be able to extract energy from their hosts. Now, to be fair, this is more complex than just one strategy. But one way that they do that is they can literally break down the host tissue and use it for energy. Okay, so to be fair, they don't always extract energy in just this way. But one way to do it is to release digestive extracellular enzymes and actually sort of rip apart the host tissue and extract energy from that and even use it as a food source. And so you can see that here on this next picture, where you've got some human cells that are glued together, and that's here called cell cement, for example. And then you've got bacteria releasing enzymes that are breaking that cellular cement apart. They can use that for energy, and in the context of infection and disease, that also allows them to spread from one area of the body to another, which is kind of what this figure is showing. But it shows them breaking down the cellular tissue here. So that ultimately has other downstream effects, but the fact that it hurts the host is obviously part of that. So some specific examples here, and then we'll move on. Mucinase. This is an enzyme that is common in pathogens that are affecting the intestinal tract. Your intestinal tract, as I'll show a figure here in a second, is lined with a lot of mucus, and it's a protective barrier. Pathogenic bacteria can break that barrier down and then actually get into the body given other conditions. Keratinase is an enzyme that actually breaks down your skin and your hair. So this is one that microbes on the outer layer of the skin can use to kind of burrow down into the tissue sometimes. Collagenase is another similar enzyme to keratinase. Keratin and collagen are both parts of the skin and connective tissue. So this is an enzyme that helps break down another layer of the skin. And then another example here is called hyaluronidase. This is actually the one that breaks down that cellular glue that I showed you on the last slide there. And so this is something that kind of helps them move through different types of tissues. You might think of these almost like a toolkit. And pathogens are kind of like robbers or criminals who have certain tools designed to facilitate their crimes. And so one of the things that differentiates pathogens from saprobes is that pathogens tend to have toolkits that are designed to do harm to living things. Saprobes tend to have toolkits that really are designed to simply break down things that are already dead and therefore not really a problem. But as we said here, just to go back real quickly, you have that category of facultative pathogens, facultative parasites, that can actually do both. And so in that situation, you have more of a complexity where they actually have a more diverse toolkit. So they actually have tools that can allow them to survive in the environment, but then they can switch and adapt with other tools that they can use if they get the opportunity inside of a host cell. So these are some of the more opportunistic survival strategies that are used amongst pathogens. Okay, this is a figure here showing a illustration of the intestinal lining. This would be like your small intestine, the cells that make up some of the villi and the microvilli in your intestinal tract. And what is usually external to these cells is a thick layer of mucus. And bacteria are often impeded by that mucus. But certain bacteria, like Clostridium difficile is the one this is shown here, can actually break through that mucus and then burrow down into the lining of the stomach and cause inflammation down in there as well. Helicobacter pylori, H. pylori, is another one that would look a lot like this here, doing the same thing. So microbes that have that mucinase enzyme are more likely to cause intestinal issues because they can break down the barrier of mucus that normally keeps them out. This is another slide here kind of elaborating on that. The truth is there's always more to it, and as this slide alludes here, you can pause it to look at it more, is that your diet actually plays a role in this as well. And so what this is actually showing is that depending on the diet that you eat, you can make it easier or harder for the microbes to break through your mucus. And so what they actually find is that mucus-eating bacteria are more likely to invade the tissue given that you have a poor diet that doesn't have as much mucus-building structures in the food itself. Okay, so in this next part, we're going to look at how these things kind of all come together in the environment. And so what are the factors that are going to make it more likely or less likely that an organism is able to actually do all the things that it is supposed to do, whether that's a saprobe or a facultative parasite or an obligate parasite, etc. The environment is going to ultimately be the ultimate dictator of whether the organism survives or not. And it's important for the organism to have its genetic variability, the ability to make and decompose matter. But at the end of the day, you either have the right environment and the right building blocks or you don't. So the first thing is substrate availability, both macro and micronutrients. Do you have the right nutrients or do you not? The next thing is the enzymes produce your genetic variability. As I mentioned before, what is a growth factor? What can you make on your own? That varies from one organism to another. Outside of those two things, the next biggest factor tends to be temperature. Temperature is something that really either speeds up the molecular pathways that we'll get more into in the next section or slows them down. And as a result, temperature ultimately sets the speed at which life can function, and some organisms can only do the functions at the right temperatures. Oxygen is an interesting one. We'll talk about that here in a minute, and then pH as well. So these are going to be the major factors affecting the survival of an organism in the environment. Now, once you have an organism with all of those factors in play, the next thing that's going to happen is that the organism is going to have to be able to get the nutrients into the cell. So at the microscopic level, cells have transport mechanisms to bring those key macro and micronutrients into the cell. We just talked earlier about how the digestion takes place, but here we're going to look just in real minimal detail about how these things can kind of get across the cell. And this touches back on what we talked about with cell membrane proteins earlier in the semester in Unit 1. So first of all, diffusion is going to be from the textbook, but make sure you're familiar with the definition of diffusion. I actually don't have the definition here, but you want to be familiar with what that is. So diffusion is the movement of molecules from an area of high concentration to an area of low concentration. And there's similar definitions to that. So when it comes to diffusion, some things can actually just move across the cell membrane from their natural high to low concentration gradient. The only thing that usually stops that is selective permeability. The cell membrane of itself is kind of like a gatekeeper, and certain things can move right across, but certain things can't. So small non-polar molecules like oxygen is something that can move right across the membrane. Larger structures cannot necessarily pass through the membrane. Some of these require what's called facilitated diffusion. Facilitated diffusion still takes place from a high to low concentration, a natural concentration gradient, but requires a protein in the membrane to move the substance from one side to the other. But in both the situations, the concentration gradient from high to low is what allows the molecules to move either from the outside of the cell in or the inside out. Now, for for nutrients or substances that are required in a manner that is outside of their natural concentration gradient, you have active transport. So active transport will usually move things in large volumes and or against the concentration or diffusion gradient. So if, for example, we have a high concentration inside the cell and a low concentration outside the cell, but we still want to move something into the cell, that would be going against the concentration gradient. You'd be attempting to go from a low to a high concentration. In order for that to work, you need active transport. You would actually need to use energy along with the membrane protein, similar to facilitated diffusion, the difference being that it also requires energy. And the energy is required to move it against the concentration gradient. You can think of it this way. Diffusion is sort of like moving down a hill. It requires virtually no energy, whereas active transport is like going up a hill, and it requires energy to go up a hill. And diffusion can also be referred to as passive transport. So there's actually passive transport, no energy, active transport, energy. These are some figures here from your text showing some of the membrane proteins that move substances from one side to the other. Some of these alter the molecules as they go. And then this here is shown as a way for larger substances to be engulfed by the membrane and stored in vesicles, whether that be particulate matter in this process here called phagocytosis, or liquid matter in a process called pinocytosis. So these are examples here of active transport. Okay, this summary table here goes over passive and active, and you should be able to find this. This is from the previous chapter version. I apologize. You'll have a table similar to this now, chapter 9. So you can use that as a reference. Okay, temperature, as I mentioned, is a big factor. So real quickly, temperature adaptations include sort of different types of organisms here, but they all kind of take place within sort of an optimal temperature range or somewhat outside of that. So you have the minimum, which is the lowest temperature that you can survive at, maximum, the highest temperature that you can survive at, then you have the optimum, which is basically the temperature that you thrive at. Okay, so kind of like humans, we can survive at pretty cold temps, we can survive at pretty hot temps, but we tend to thrive somewhere in between the extremes. And every organism has its own optimum, and that range is sort of where it tends to be at its best. So to put that into some examples here, some microbes have a very narrow temperature range. You see this with certain parasites like the rhinovirus, which is a common cause of upper respiratory infections, very small degree of temperature fluctuation. Some pathogens like Staphylococcus aureus we mentioned earlier, which is a facultative pathogen, facultative parasite, it can survive and function from as little as 6 degrees Celsius all the way up to 46 degrees Celsius. Enterococcus faecalis, which is a common intestinal microbe, similar, even slightly larger range there. So the truth is, temperature adaptations are all over the place. Some have very narrow, some have very broad. But you can kind of put them in 3 distinct categories. And it's not always quite so easy, but there's sort of 3 general categories. So a psychrophile is a cold-loving organism. That number is usually right around 15 degrees Celsius. Mesophile, moderate temperature, 20 to 40. And then thermo means heat or hot. So a thermophile is a heat-loving, hot-loving microbe. So you're looking at 45 degrees Celsius and above. So again, it's not always quite so obvious to put these in terms here, but ultimately you have 3 general categories that you can sort of think about these in. Okay, so temperature. The next one is oxygen. So you and I as humans, we don't have to think about oxygen because we just require it and we have to breathe it, and it's usually something that we don't think about. So we are aerobes. We use oxygen and we require oxygen, and as a result, our body is able to use oxygen. Now technically, we're actually an obligate aerobe, cannot grow or function without oxygen. When you look at organisms, however, not all of them require oxygen. Most of the ones that we're familiar with do, animals, plants, but when it comes to microorganisms, especially bacteria, there are quite a few of them that are anaerobes, and they actually don't require growth, does not grow in oxygen, does not require oxygen to grow. One of the things you find about oxygen is that oxygen tends to be harmful. And so as an aerobe, as we'll see on the next slide here, you actually have to be able to sort of neutralize oxygen damage. Aerobes, when it says lack enzymes to use oxygen, using oxygen is kind of like playing with fire. And if you do that in the wrong conditions, you'll get burnt. So our ability to breathe oxygen is dependent upon enzymes that we have to actually almost like put out the fire that is caused with aerobic respiration. We'll get to that on the next slide here. So aerobes require it. Obligates cannot grow without it. Aerobes don't want oxygen, in fact, are often hurt by oxygen. Facultative anaerobes are kind of like facultative parasite. That term facultative comes up again. It's kind of either or. And these can utilize it if oxygen is available, but are also capable of functioning without it like an anaerobe. So they're literally kind of both. Then you have a few other subcategories here. Aerotolerant anaerobes is one of those that do not really utilize oxygen, but do have the ability to minimize some of the harmful side effects of it. Okay, and there's a few other ones like microaerophiles that I don't have listed here. But you have essentially four main categories here: aerobes, anaerobes, facultative anaerobes, and aerotolerant anaerobes. So aerotolerant anaerobes are like anaerobes. They don't use it, but the difference is these can't survive in the presence of oxygen, and these can usually survive in the presence of at least a little bit of oxygen. So what is it about oxygen that's so harmful then? Oxygen is metabolized in your cells in a way that produces oxygen-free radicals. And oxygen-free radicals are something that our body then has to neutralize in order for those free radicals not to cause harm. And I kind of think of them almost like baseballs bouncing around in your cells. And if you have baseballs bouncing around in your house, they're going to cause damage. And so every time oxygen is metabolized, a baseball potentially starts shooting out all around the house, which is the cell. At that moment, you need to immediately grab that baseball and capture it and prevent it from shooting all around your house, bouncing off the walls. If you can't do that, then those baseballs, these oxygen-free radicals, are just going to continue to destroy the inside of your house little bit by little bit. And every time you metabolize more oxygen, you produce more of these free radicals, like having more baseballs bouncing around the house. And so you simply can't have all of these things bouncing around causing damage, and so you have to neutralize those. And ultimately, your cells, although it's far more complex than what we're saying here, your cells ultimately have enzymes that neutralize these oxygen-free radicals. And your body can also produce compounds called antioxidants. Antioxidants can help with oxygen-free radicals. Certain foods like blueberries and raspberries are known to have high concentrations of antioxidants. And it's believed that one of the reasons why those are good for you is they help with the oxygen sort of mitigation or neutralization. It's somewhat up for debate how much eating blueberries helps, because the truth is your body makes its own antioxidants. It's certainly good to eat them. I think blueberries are great, but how much you need it from an outside source is up for debate. Your body makes antioxidants, and theoretically you can neutralize them all on your own. That's a whole other concept there, but that's the idea of why oxygen is actually harmful and why not everything can live with oxygen. And these are some examples here of some of the byproducts that your body makes that are harmful. So that's where oxygen comes in. pH is a matter of acids and bases. And we'll keep this one relatively short, but acids and bases can be close to neutral or strong or weak. The truth is, most of the time our pH falls between 6 to 8, which is very close to neutral. Neutral is 7. However, anything outside of that range has a greater potential to damage our cells, and ultimately strong acids would be 6 and below, strong bases would be 8 and above. And although each acid has its own characteristics, generally speaking, either the lower the number or the higher the number below or above below 6 or above 8, the more likely those acids are to affect you. What they ultimately do is they cause denaturation of enzymes, and they damage cellular structures such as the cell membrane or proteins located on the cell membrane itself. If they get into the cell, they can even damage things like the DNA, for example. So most of the time, pH is relatively neutral, and we don't have to worry about it. But when we are exposed to strong acids and bases, that's what can happen. They damage our cells. And the last factor that is a lot more complicated is the ecological associations. The fact is that microbes rarely, in fact, almost never, live in a isolated existence. They share associations with many other microbes, whether that be microbes themselves or even multicellular organisms. And as we mentioned some of these terms earlier, these are usually symbiotic, or they're at least often symbiotic. And we mentioned a mutualistic symbiotic relationship earlier, which is common among saprobes. And then we mentioned parasitic symbiotic relationships, which is common in human pathogens, for example. Commensalism is sort of in between. And commensalism would be where one organism benefits, but the other one doesn't, or is not harmed, at least in the process. Sort of the difference between a commensal and a parasite is that the parasite causes harm at the expense of the host, whereas a commensal wouldn't cause harm but may not benefit at the same time. And then you have some non-symbiotic relationships. So synergism would be where they're not really required, but you can benefit having other microbes around you. Kind of like you might have a better experience having other people around you, your friends, your family. You'd feel better with certain people around you. And then the opposite, antagonism, where you actually are worse off. You're not necessarily inhibited or hurt necessarily, but you just don't do as well as you would with a synergistic relationship. And the last thing here is, we talked about this before, but the normal microbiota in the human body is an example of really both mutualistic symbiotic relationships and commensal symbiotic relationships, and those relationships can become parasitic if we don't take care of our bodies, or if we're exposed to certain pathogens, or both. And so the normal microbiota is this example of this complex interaction between different kinds of organisms all inside the body, and represents really a multitude of different kinds of relationships. But the microbiota is really mutualistic largely, but more complex than that. But they ultimately help remove skin, aid in food digestion, ward off bad bacteria, produce certain vitamins, maintain pH, and even more than that. That'll be a topic more in the last unit of the semester, when we start with the human microbe interactions in Unit 3. All right, we'll stop it there for today, and we'll do the metabolism chapter in our next section.